Thank you so much. I'm so excited to be here. It's my first time in Brazil, and it's been a wonderful welcome, and everyone has made me feel so welcomed. There's a QR code here, and then scattered throughout the talk with a link to the slides. I tend to talk too fast, and so then if you miss something, at least there's a QR code if you want to be able to come back to it later. Hopefully I saw lots of pictures of things at the poster session, and the posters have been really fantastic. So I hopefully don't have to convince too many people that in addition to biological images being pretty, which they are, that they're also informative. And we're really passionate about doing stuff with images because even compared to other omics, which have advanced so much in the last 20 years or so, biological images, which we've had for 400 years, are still, in a lot of ways, the best way to get data about a lot of phenomena. You can get information about single cells, you can get information from live cells, you can watch things over time, and most of these you still can't really do with other omics. Or if you can, technically, you can't really in a way that's sort of cost efficient, and you can do it more than one or two things. But when we look at biological images, we sometimes lose the trees for the forest. So digital images are ultimately just arrays, and that's true, for example, every time you snap a picture on your phone to upload to Instagram, or this picture of a man taking a picture, these images that we take are just arrays of numbers, which means that we have tons of quantitative measurements, and so they're actually a really rich data source. If your microscope has a 4-megapixel camera, that's 4 million data points every single time you snap that camera, which you might do for multiple time points or z-planes or channels. So we actually have a tremendous amount of information that we can get from biological images, and I'm going to really talk about that more later, but first I'm going to talk about how we actually find objects in things in the first place. Unfortunately, people who spend most of their time making images often aren't trained to think of image analysis as part of the cycle of doing biology and doing microscopy. We sort of think that sample preparation, image acquisition, and then stop, and then you do that a bunch, and then eventually you go in and you do some analysis. But unfortunately, if you do that, by the time you start doing your analysis, there might have been something that you could have figured out if you'd analyzed your pilot quantitatively, that you could have fixed something in the sample preparation and the acquisition. You could have picked a different dye or picked a different microscope such that your analysis would have been an awful lot easier and you would have been able to get more reliable data. So these are the things that underlie the imaging platform, which was founded by Ann Carpenter, who's, of course, one of the original creators of Cell Profiler. We write software that turns images into the answers to questions. So you might not think you need, you're in this room, so you might at least think there's a chance that you might need to quantify your images. But even if you think that you don't, or even if you think for a lot of your phenotypes you can see them just fine, human brains are great at many things, they're not great at quantification. So if you're sitting in the back, this might still even look to you like a color picture. This is not a color picture. This is a grayscale picture with some colored lines drawn on top of it. And your brain helpfully fills in the missing information because your brain is just trying to give you a quick picture of what's going on around you. But it's not giving you accurate data. So you shouldn't trust your brain to tell you quantitatively what's going on in a picture because your brain is simply not built to do it. And so most of what my lab does is around measuring and scoring known phenotypes. That's what I'm going to spend the majority of the time talking about. But I will then talk just a little bit about more unbiased machine learning things sort of right at the end. So I told you we have these big arrays of data. We have millions and millions of pixels. How do we actually tap into those things? I mostly just show this slide because I want to sort of point out the really fantastic group of folks who over the years have worked to make Cell Profiler work and be usable for lots of people. And I think one of the reasons that Cell Profiler has sort of stood the test of time, Cell Profiler turned 20 last year, which means it's old enough to drink this year in the US. That since the beginning with Ann Carpenter and Ray Jones, there's always been at least one person on the team whose original training is in computer science, which are the folks in blue here, and whose original training is in biology, which are the folks in green, and including me. And so that means the Cell Profiler has good software engineering practices, but the documentation, for example, has all been written by biologists for biologists. So we try to make it and put things in ways that biologists are going to know and understand. I'm not going to, this is totally a Cell Profiler talk, so I'm not going to belabor this too much. But this is the Cell Profiler interface. There's really nothing in Cell Profiler that you can't do in, say, ImageJ. Who here has used ImageJ? Yeah, that's what I thought. What Cell Profiler does allow you to do is if you don't know how to code or you don't feel like coding right now, Cell Profiler is ultimately under the hood an image analysis workflow tool. It lets you make a pipeline where you can put modules together in any number or combination that you want, and it makes a reproducible, reusable workflow that you can apply to however many images you have, whether that's 1 or whether that's 10 million. And that workflow will actually even be saved as part of your quantitative data unless you turn that off. Please don't turn that off. But you can then put together all of the modules in the pipeline, you can configure each one, and then you can use it to sort of analyze just about any kind of biological data that you have. This is the most important button in Cell Profiler. This is the Help button. There's about 100 modules in Cell Profiler, and across all of them, there's about 1,000 settings. We don't expect you to memorize all of that. I've been using Cell Profiler for 15 years, and I still forget some things that it does. So this Help button will allow you to find out whatever a particular module does or what a particular setting does so that you don't get lost. Once you have figured out the right workflow, you can go into test mode. You can start applying this pipeline to sort of one image at a time. Do it to your negative controls, do it to your positive controls. You did some controls, right? And check to make sure that it works the way that you want it to. And then once you've checked on your controls, you set your output setting, where your output folder wants it to go, and Cell Profiler can then just analyze the data multi-threaded up to as many cores as your computer has, and you can go off to get a coffee or a beer or whatever else you like to do rather than sitting there counting cells one at a time. So how do we do this in practice? So when I teach Cell Profiler workshops and I start teaching people how we do segmentation or finding objects and images, this is where we start. That's a lot of slides. That's a lot of information. And maybe you want to become a bioimage analyst, and I totally recommend that you do if that's what you want. But maybe you just want to find the answer to a biological question and then go back to your life. So thankfully, there are some better things coming that, again, really 5 or 10 years ago seemed like science fiction, but are actually now helping make this process of bioimage analysis a lot easier. One of the good things about ChatGPT is I no longer have to ask people if they've heard of deep learning. I think most of you have now. It's not just for ChatGPT. It is also for doing things with images, which you might have found out if you've played with things like DALL-E before. And it has a lot of uses in bioimaging. You're going to hear a lot more about this later. But so some of these are things from our group, some of these are just things that we're fans of. But you can do things like virtual staining. What would this stain have looked like if I added it to my cells? I didn't, but I want to know what it would have looked like if I had. Or, this week my secondary antibody is a little bit dimmer. Can you make it look like last week when it was brighter? And so all of these are things that are possible, and I'm happy to talk to folks about when you might and when you might not want to do some of these things. But really, the place where it's come the furthest in biological imaging is in segmentation of objects. And so I just want to call out a couple of things that if you're new to bioimage analysis of light microscopy images, you really ought to know about. Stardust is a really popular object finding thing for 2D and 3D. We have a plugin for that in Cell Profiler. CellPose is my absolute favorite neural network. This thing is basically magic for finding objects and images, including in bright field images. If you haven't tried it, try it. We have a Cell Profiler plugin for that also. Here's the problem with all of this deep learning stuff, though. I know, I just said deep learning fixes the problem, and now I'm saying there's a problem again. I'm terrible. When we do surveys and we ask people how much of their time they spend making images versus analyzing images, and what their computational comfort is in developing new computational skills, you might not be surprised that people whose day job is working on their laptops are more comfortable developing new computational skills than people whose day job isn't. And so people who spend most of their time making images just don't tend to be super computationally comfortable. We have some people who are, but the vast majority of people really aren't. It's not their day job, it's not what they're trained in. So the person who is making the deep learning model might be very different from the person who is the expert on what the deep learning model is supposed to find, and that's a problem. It's especially a problem because installing scientific software anyway, I'm sure most of you have found, is terrible. It's really terrible for deep learning. And so these are just a few things from the image.sc forum, which I'm going to mention several times more. This isn't just that my team is really bad at their job. My team is really good at their jobs. This stuff is just hard. We recently looked through the image.sc help forum, which is a help forum for more than 60 different image analysis software tools. And the word install comes up in the first post of 1 of 10 threads. It comes up in a thread 1 out of every 5 threads. So this is a huge problem. If you can't get the software to run, you're never going to actually get it to answer your questions. And this is only going to get worse with deep learning. So what can we do? Well, one of the things we can do is we can put all of the software on the web. That means that the problem is for people like me who make the software and not people like you who use the software. You never need to install anything because you just visit a website and the software works. That sounds better, right? Yeah, that one's better. And so there's a lot of tools that are sort of coming out and are becoming available where you can actually start doing image analysis on the web, and I definitely recommend you try any or all of these. ImageJ.js is ImageJ but in the browser. Since all of you know about ImageJ, you can now just do it in the browser. ZeroCostDL, if you're comfortable using Jupyter notebooks, this is a great thing to do. And Galaxy is a way for you to run image analysis workflows much like Cell Profiler, and including Cell Profiler, but on somebody else's hardware where they've already done the work of installing your tools for you. But I want to tell you about one that we've been making. We've been working on this for about 7 years, and it's finally done this year. Well, software's never done, but this is Beth Cimini. You can find it at bethcimini.app. And if I see you guys on your phones or your laptops for the rest of the talk, I'm going to assume it's because you're going to bethcimini.app. That's what's happening. So Beth Cimini is an in-browser image analysis, images to discovery workflow tool. Our idea was when we started making this that you would be able to upload an image, find the things you care about, measure the things you care about, and answer all your questions without ever having to leave your web browser. And what makes Beth Cimini a little bit different from these other tools that I mentioned, or at least some of them, is all of this compute is happening on your local computer. When you put images into Beth Cimini, you are not sending us your data. You're not sending it to a central server. We don't know about your data. We don't need your data. As soon as you go to the website, you can put your computer in airplane mode. It will work exactly the same way. This is all happening on your local machine. And so if you have concerns about things like human subject protection, or there's just reasons you don't want your data going up on the internet, this is a way you can use a web tool but not actually send your data anywhere. Because we want you to be able to train segmentation models where you can find things, or you might just want to sort of find things by hand, we have a really great annotation tool that's part of Beth Cimini. Trainable segmentation isn't in there yet, but we're hoping by the end of this year. But in the meantime, the annotator works and is really nice, and you can download these annotations and you can save them and send them to other people, and they can edit your annotations. This is the SuperPixel tool. Oh, I took out the one of my favorite, which is the color annotation tool. Try the color annotation tool, it's fun. And here I'm going to train a classifier. Movie's going to happen in real time between cells with no GFP, cytoplasmic GFP, and nuclear GFP using deep learning. This is real time. So we pre-labeled about 60 to 80 examples of each class, so this didn't take much time to actually say which is which. And we end up with a classifier that's 90% accurate in about 30 seconds. So this is going to give you the power to do this kind of deep learning, but again, on your local machine without installing anything. It even works on phones. I've been told by my software engineers to stop showing it to people at bars on my phone, but you can't actually do this on your cell phone. And the last piece, which is still in the beta version of the app, but that really sort of takes us from images to discovery, is we're now finally have pulling in measurements as well. So you can take those same cells that you just classified as neo-GFP or nuclear GFP or what, and you can also measure, say, their size to see if the ones that have nuclear GFP are, say, bigger because that's caused cell cycle arrest. All of that can now happen inside one tool. And this is really key because measurements are ultimately, most of the time, what we care about. Sometimes you just care about counts of things, but often we want to know the size of something or the intensity of something. And so measurements are ultimately at the heart of what we're doing when we're doing image analysis. So Cell Profiler, for example, has tons of measurement things that are built into it. Piximi only has a couple so far, but we're working on building out more. And again, many of these metrics can be found in lots of tools. If you like R, if you like Python, if you like ImageJ, if you like Cell Profiler, you can do them however you want. And the ones that, as biologists, we're used to thinking about are things like intensity or things like size and shape. Those are the things that we're used to thinking about. But remember I started with, you shouldn't trust your brain because your brain is bad at this. These are images of cells in different cell cycle phase. They were taken off an imaging flow cytometer. Who wants to come up and say which one is which? Only one time has anybody ever raised their hand for that. I was really impressed with that one person. You might have guessed that these are in order, and certainly by the end of here of the cell cycle we can tell, but if you scrambled these ones up, I certainly wouldn't be able to unscramble them, and you probably wouldn't either. But a computer can do it really accurately, just from information that is like subtle textures and granularities that we literally do not see. That information does not go into our brains, but a computer can do it just fine. And even the mistakes the computer is making, it's not confusing, G1 for metaphase. It's saying that S and G1 are a little bit similar, but that's not a perfectly switch-like transition either. So the mistakes are just as much sort of about our forcing the biology to be in rigid bins. Biology doesn't like rigid bins. Biology is messy. So there's information here, like textures, that make no sense to human brains, but a computer can use it. So that's a little bit why I'm saying we should take some measurements that don't make human sense, because for some tasks like this, the textures are things we literally don't see, but they are useful. And so that's going to lead me into the next to last part of my talk, which is about the work that our sister lab, the Carpenter-Singh lab, does around making more out of your images by measuring things that you never thought to measure in the first place. And so this field of image-based profiling, which if you attended one of the morning sessions, you've heard some about, the idea that essentially, now that people are used to things like single-cell RNA-seq, people are more familiar and comfortable with this idea of we're going to measure a crap ton of stuff, and then we're going to cluster it, and we're going to see what falls out. But you can do this with images, too. You can measure hundreds or thousands of image features, and you can cluster it, and you can see what falls out. And so about 15 years ago at the Broad, my boss, Ann Carpenter, and some other folks, including Stuart Schreiber, said, can we do this on a microscope by trying to find the cheapest possible dyes that we can? They're not necessarily completely cheap, but the cheapest ones that you can sort of get that work at scale. And can we just image the maximum number of organelles at a time so we can get the most measurements at a time? And again, most of these are dyes that are pretty common. They're not anything wildly exotic or crazy. And so even in ways that the human eye cannot detect, so these are cells that are sensitive to a particular proteasome inhibitor. These are cells that are resistant. I can't tell the difference. I've spent my entire career looking at pictures of cells. I can't tell the difference. A computer can do it with really high accuracy. And so you can imagine the power of being able to do things like tell which drugs a cell is resistant to with an assay that is only a couple of US dollars at scale. And so we're really excited about approaches like this, because again, since the dyes are just for compartments and not for particular kinds of biology, you can measure lots of kinds of biology in the same assay. And so we're working to combine this with other omics. This was a fun thing where we had iPSCs from many donors, and we did cell painting and single cell or whole genome sequencing in the same iPSC donors, and then we said, can we actually connect genotype to phenotype? Spoiler alert, we can sometimes. When you have 3,000 features and thousands of genes and 300 people, it turns out the curse of dimensionality, it's hard to get enough statistical power to say anything reliably, but we found a couple of genes where we actually can say that a human variant of unknown significance actually is causing a particular change in cell painting. So hopefully what you're thinking at this point is not how much longer is left, it's about 4 minutes, but oh wow, this was all cool. I want to learn how to do this. Where can I go for help? So for our individual tools that we make, we have lots of in-app documentation. Again, here are all of those help buttons that I mentioned in Cell Profiler for all of the different settings in this particular module. It's a lot of help buttons. And for Cell Profile in particular, again, for a tool that's 20 years old, we've spent a lot of time building up examples and tutorials where you can say, all right, which of these things looks kind of like my data, and try to pick the one that sort of most closely matches. Both of these tools also have manuals that you can go and you can look up a lot more information. But assuming you're not using our tools, there's still some good resources for, no matter what you're doing, getting help. So bioimagingguide.org is something we put together with Bioimaging North America last year. And the idea was to create a website to teach you what you don't know about bioimaging and bioimage analysis. So if there are things that you don't know you should do around sample prep or around imaging or around analysis, where is a place where you can find information about what you should go learn? And then we have links to resources of where you can go learn it. Our idea is not to make this a 1,000-page textbook, but to make this a thing where we can point at the very best examples of educational materials. And because we made this in a technology called Jupyter Book, it's on the web, yay web. We can do fun things like drop-downs. We can put notes in the margins so that, if there's something that you want to learn a little bit more, but otherwise if you don't care. But one of the really exciting things about this Jupyter Book technology is it's very easily translatable. So thanks to the wonderful and talented Mario Cruz, who you'll be hearing from in just a bit, you can now read this entire book in Portuguese. So thank you, Mario, for that. Globius is the Global Bioimage Analyst Society, and is a new society that's just started in the last couple of years. It has things like events, training, and education portal. You can find out events. Find your local bioimage analyst. If you are a bioimage analyst, please add yourself. If you want to find a bioimage analyst, please go look for one, and you can find help that is near you and with expertise that you need. And I mentioned the image.sc forum. There's many, many tools, and you have a lot of experts from a lot of different places. So that the idea was that you can just post your picture and your question and say, here's my image, here's what I want to know, help. And you'll have experts from 60 different tools who'll say, oh, this is how you would do it in ImageJ, and somebody saying, oh, well, this is how you would do it in Cell Profiler, or you shouldn't do this in this tool, you should do it in that tool instead, and you'll get really friendly answers from all over the world. It's incredibly friendly. We really love when beginners post on there, so please, please bring us your questions. We're all nerds who love talking about this stuff, so we really actually want you to bring us your questions. And for my team's tools, my team does office hours every 2 to 3 weeks. They're open to the general public. Please sign up. And you can just come work with us. We collaborate with groups all over the world, and we're really happy to figure out ways to collaborate with anybody here. And so that's my time, so I just want to thank the awesome people in my lab and in our Carpenter Singh lab who've done all this work. QR code for the slides is there, and we have time, I hope, for a couple questions.